last year there was a really interesting study. It was a battery benchmarking study where they just asked 21 groups around the world to take the exact same materials from the exact same suppliers, build a battery and test it. Those were the only instructions. And the data they got is hugely variable. You don't have to know anything about batteries to just know that if you're starting with the exact same materials and you're supposed to be building the same thing the same way, you shouldn't have this much error, right? So why does that happen? Everyone had the same information, everyone had the same inputs, in fact. But something between that input and that output differed. And one place that we often look when we talk about data science is to bioinformatics, because the reason that AlphaFold, which is kind of one of the gold standards for using big data sets, was able to be developed is because of the open source and consistent deposition of data in the protein data bank. And as we think about that, what we need are similarly consistent metrics for CleanTech. And so that's what people are trying to do with batteries right now. What we're working towards is what's called a battery genome. So this would effectively be consistent input data with very specific metrics for how these tests are run, so that we have a data set that is as complete as the protein database, but for battery materials. So batteries are basically at the point now where photovoltaics were probably 10 years ago. And so now we have enough data to actually build a database. But what about newer technologies? So one thing that my lab focuses on is electrocatalysis. And we think about this basically in terms of very simple transformations that you can do with water and carbon dioxide. And the reason we focus on those is because if you take water and carbon dioxide and use clean energy inputs, you can then use electrochemistry to convert that to either chemicals or energy, which then, even if they're generating CO2, such as some of the fuels you're making, are at least circularizing the carbon economy. And so one way that my lab approaches that is by taking inspiration from nature. So pretty much since humans have been building things, we've been looking to nature for inspiration. So if we look at the top right, that's a suit of armor from the 1200s, and it was inspired by the scales of the pangolin. Does anybody know what this black and white image is? You could just yell it out.
Tentacles on a shark?
Close, yes. So this is actually swimsuit material from the 2010 Olympics. It's since been banned because it gives swimmers too much of an advantage, and it's based off of shark skin. How about this one? Velcro, excellent. Velcro, something we think of as a recent invention that is completely of the human-built environment, is actually based on the interaction between seed coatings and animal fur, which is what we see on the far right. And that interaction is basically what the hook and loop was designed based on. So even things that we think of as completely human-derived are actually nature-inspired. And so what my lab focuses on is thinking about certain types of reactions in biology and how we can harness those. So another quiz for you. For those of you who haven't seen Family Feud, normally you call out top answers, so we're all just going to call out top answers. So when we think about chemical reactions in biology, what do we think are some of the most important reactions?
Photosynthesis.
Excellent. So we have photosynthesis. Any others?
Decomposition.
Decomposition, excellent.
Nitrogen cycle.
Nitrogen cycle.
The water cycle.
Mm-hmm. What about how we breathe? Oxygen respiration, excellent. So I think we've gotten pretty much all of those. So when we think about these reactions in biology, we have photosynthesis, which takes CO2 and sunlight and basically makes oxygen, and water as a byproduct. Nitrogen fixation takes nitrogen in the atmosphere and fixes it as ammonia, which helps plants grow. Respiration can either be aerobic, where we're basically generating energy, or fermentation. These are what happens when we don't have oxygen available. So what do all of these processes have in common?
Can you go back to the last slide, please?
Yeah,
And the microbes?
they're done by microbes, that's great. What is moving in these reactions?
The input is output.
Yeah, which is basically electrons, right? These are all redox reactions. So these all involve electrons moving. And the easiest way to both control that and to probe that is electrochemistry. So that is why we think about electrocatalysis. And so what I'm going to talk about are some of the projects in my lab now. Stepping away a little bit from data standardization, but mostly just to show how we can use biology for sustainable technologies by having this fundamental knowledge about the processes that they do. So one of the main areas of research in my lab is how we can make cheaper protein materials. So has anybody in here had to purify a protein before? I see a couple nods, excellent. So I was a very lazy postdoc and I didn't want to have to purify proteins. So what we developed was a way to basically tell cells to put the protein on the surface. And so now we have materials that are basically a cell with the protein. And why we care about that is that enzymes make up a huge part of our chemical synthesis market. They are an $8.5 billion industry currently, ranging from extensive use in pulp and paper processing to chemical manufacture and even in animal feed. And the problem is, as I mentioned, protein purification is awful. So the way currently that it's done industrially is you tell a cell to overexpress or make a lot of a certain protein. You can grow those cells up at scale. And then about 90% of the time, energy, and cost associated with getting a protein is busting open those cells, isolating that protein, and purifying it. And that protein is not very stable once it's outside the cell. So by scaffolding these on the surface, what we've been able to do is to make these inexpensive materials that allow us to do everything from environmental remediation to clean energy conversions to even recovery of materials and resources. So we'll talk about a couple quick examples, one of which is bilirubin oxidase. This is a multi-copper oxidase, which is basically a protein that contains multiple coppers. And in nature, it can split water. So if it's in an oxidative environment, it can take water and generate oxygen. If it's in a reductive environment, it can generate hydrogen from water. The problem is this is really unstable. And so people haven't been able to use this for actual technology applications because the protein falls apart. So what we found is when you take the protein, it can do some oxygen evolution. This is basically electrochemistry showing activity. But when we take that surface expressed protein, which is about 1/10 of the effort to make and a fraction of the cost, it's 70 times more efficient for oxygen evolution. So you're saving time and resources and getting something that's more efficient. And because of this increased efficiency, we decrease the amount of energy required to generate oxygen by about 35%. And so that's just one example. Another is rare earth elements. We've been hearing a lot about these because of limitations on imports of these materials. They're used in pretty much everything from cell phones to sonar and radar systems. So they're basically essential for our daily lives. The problem is currently we recycle less than 1% of these. And the way they're actually mined is what's shown here. You dump a lot of very strong acid into a hole in the ground and wait for that to dissolve minerals that contain rare earths. The problem is when those ponds leak, you get things like this, which is basically acid drainage. And that destroys ecosystems around those mining sites. And so what we want to do is develop inexpensive materials that are not only inexpensive and easy to use, but are more sustainable. And the great thing about nature is it's already evolved things we can use. I tell my grad students that using biological systems basically gives you a 4 billion year head start on your Ph.D. So there's a protein that can bind rare earths. It binds 3 rare earths with parts per billion affinity. So sub-micromolar, nanomolar affinity. And it's pH sensitive. So we can bind these and then release them. And what this means is we can make things like filters, which is shown here. We get about 83% overall recovery over 10 cycles. This is in the presence of 1,000-fold excess of other ions that are usually present. So this is things like calcium, magnesium, zinc, and aluminum that we don't want to capture. And what that means economically is that now, instead of having to do this whole process, we skip that whole downstream part. So this requires no specialized equipment, it's simple to scale, and it's less than 10% of the conventional cost of proteins. What this means for things like recycling is it costs us about $10 a kilogram to make these dried cell materials, and we can recover about 12 milligrams of lanthanide per round of leaching. And so that means we have a cost of about $150 a kilogram for rare earth element recovery. Why we actually care about that is because the market value of rare earths is around $2,000. And so this is an actual motivation for companies that currently aren't recycling their rare earths or e-waste in general to actually start doing that. And so as we think about how to expand the application of proteins, we also want to think about what we can do with whole cells. And so we were able to actually combine technologies where we can use whole cells as well as these protein materials to both sense and destroy environmental contaminants. And I thought this would be an interesting story to talk about because of how we've applied this to autonomous sensing by integrating these into RFID tags. So the specific type of cells we're going to use are very cool microbes, in my opinion. There's a whole class of microbes that can basically spit out electrons for respiration. These are things like cable bacteria that are shown, or geobacter, but the ones we use the most are called Schionella onadensis. They were isolated from pond scum in New York. This is Lake Oneida, where they were isolated from. And these guys are genetically tractable, which means we can engineer them. And the way they actually spit out electrons is through this pathway of proteins. Each one of these contains about 10 heme groups, which basically makes a wire across the membranes. And so this allows us to engineer these and use them. So now we're going to change back to those protein materials again and talk about pesticides. So organophosphate pesticides are about 50% of the pesticides used globally. And they're terrible for agricultural workers because they chemically look a lot like and act a lot like chemical warfare agents. So there are about 11,000 acute deaths annually because of exposure. And what we are finding now is that low dose exposure over years causes everything from infertility to higher rates of cancer. You can see here that these guys have great personal protective equipment, PPE. Most agricultural workers distributing this don't even have masks. So they're exposed to very high rates of this every time they distribute. The benefit of this, though, is that what you get when we degrade this is this yellow compound, whoops. So we can detect that with UV-Vis, which is just absorbance spectroscopy. And as I mentioned, what we can do is put a protein on the surface of our cells that allows us to degrade this really rapidly, as you can see with our degradation profiles there. So that's fantastic, but if you notice, our limit of detection here is around 25 micromolar. That's about 10 times the lethal dose of this. I would argue a sensor that only detects above the lethal dose is not an especially useful sensor. So we turn to synthetic biology. You don't have to worry about these genetic maps here. What you care about is that if we have the degradation product, we get something green. Without the degradation product, we shouldn't get any green. The problem is we still get some green, right? That's because we get what's called leaky expression, where we're basically getting background. Fluorescence is also harder to quantify than absorbance, so we're going in the wrong direction for making a cheap sensor. So now we're connecting those two halves. We have the protein that can degrade, but we also have these microbes that can generate an electrical signal. So now if we basically knock out one of those proteins in that pathway that allows them to spit out electrons, we're cutting the wire and turning them off. If we have the small molecule, we're basically plugging that cell back in and turning on current, which you can see here. And it's quantitative, and that's fantastic for this one molecule, right? But we want to be able to generalize this. So the great thing is microbes have already evolved really, really sensitive pathways to sense things in their environment, usually small molecules or ions. And the way they do this is basically they have a sensing protein that is specific to whatever we're trying to detect. In this case, that's that blue diamond. And then that turns on or off gene circuits that help protect the cell from that, especially if it's toxic. We can basically harness this pathway and just hook it up to our electroactive microbes. So now, if we can find those sensing proteins, we can basically have those control our electrical signal. And so for known signals, we've been able to engineer sensors for all of these analytes. You'll notice most of these are for soil health indications, because what we really wanted to do was to take the burden off of farmers, especially, to have to go physically sample soil. But many of the things we want to target don't have known sensing genes. And so what we've been able to do is to develop a program that actually allows us to search a database of genes and genomes. And if you know your target molecule, it allows you to pull out potential sensing circuits. And so using this, we were able to develop about 9 new sensors for different vinyl chloride compounds. For those of you who are familiar, this is what was released in the New Palestine, Ohio train crash last year, and is still contaminating pretty much all the farmland around that area. And so with this workflow, if there is a gene that has been annotated as a sensor that will likely work for a molecule that you could search, we can now find it and pull it out. And our database will tell us what microbe it's in. So we can literally steal that gene from the native microbe and stick it into our sensing microbes. And that's pretty cool. The problem is the way we actually sense these is with this little electrochemical bioreactor. So it's a jelly jar around this big. I'm betting if anybody knows farmers has talked to them, they're not going to want these all over their land, right? This is going to make it very difficult to sample. So how do we think about a form factor that will make it actually easier to do readout? What we came up with was an RFID tag. These are basically little antenna. Has anybody shopped at like Uniqlo where you don't have to actually check out with a person? We literally use those same RFID tags. And all we have to do is disconnect the antenna. So we literally just hole punch out that microchip and attach that to an electrode that has microbes embedded in agar on it. And so now, whatever you're sensing on those microbes, if they turn on their current and connect that circuit, your RFID tag turns on. And so what this means is now you can monitor these with either a cell phone app or an RFID reader that can be put on autonomous vehicles or drones to monitor agricultural land for around $2 a sensor, rather than having to go take physical samples. These last around 6 months, and we've been able to integrate degradation tags into those proteins so it cycles. So when you have your small molecule present, your signal is on. When it goes away, it turns off, so they're reusable. And so that's one way we can use microbes, but microbes can be very valuable across different industries. In agriculture, there's a huge effort now to basically replace chemical fertilizers and pesticides with microbes. We can also do enhanced weathering, which is a fancy word for doing basically carbon capture with microbes in the soil to change the pH. Human health, many vaccines are microbial, things like the tuberculosis vaccine, as well as probiotics and living biotherapeutics as replacements for chemical medications. We can also use these for green chemistry, things like bioethanol production or pharmaceutical production. We can also use these to make plastics or sustainable aviation fuels. And even in the built environment, I showed some examples of how we can think about recovering metals that are challenging to do in a profitable way right now. We can also use these for green mining or plastic recycling or building materials. And so I'll very quickly go through a couple of examples here of how we can do this. So one of the limitations currently with using microbes for agriculture is that many of the ones we want to use are very sensitive to things like temperature, UV light, and so they're really hard to transport where we need them to go, right? So we were able to use some materials in our lab to basically protect microbes. These are nitrogen fixers at temperatures that could cook an egg. These were stored at 50 degrees Celsius for a couple of weeks. Normally, these guys don't survive, as you can see, but all of our materials-coated ones survived. And what was especially impressive is that our stressed microbes actually improved germination. So this is what farmers care about, right? So without having to have cold chain storage or chemical transport storage for ammonia, we get about 30% cost savings for shipping. We also no longer need hazardous chemical storage on site for farmers. Because we only have to deliver these one time per growing season, we get about 10% cost savings in distribution and personnel. And because we can distribute these where they need to go, we're effectively getting targeted pesticide use. So we save farmers a lot by reducing the amount that they have to distribute, about 30% overall on chemicals. And so that's just one way we can think about using microbes that are both increasing sustainability of processes, but also improving profitability for low margin farming, which can be a challenge, right? Especially with price volatility of ammonia, especially with global events. And so the last example I'll talk about is how we can think about using microbes for biosynthesized materials. Fancy word for using microbes to make things. These are all native microbes. Nothing I've talked about except for the protein surface express have been engineered. And you've probably all encountered the microbes I'm going to talk about. Many of these are either soil derived or they're the ones that make things like stalactites in caves or help grow coral reefs. And these microbes can do something called microbially induced carbonate precipitation, which is a fancy word for if there's CO2 dissolved in water or in a humid environment, these guys can take a calcium or magnesium ion and stick it together to make limestone. And so what that allows us to do is to make very strong building materials using microbes. What you see here are just the particles after overnight incubation. And on the right, what we see are the microbes stained in green and the particles in red. And what this is showing is that those particles are not inside the cell. So that means we can get very large particles on the millimeter scale very quickly. And importantly, for construction, we can control both the crystallinity and the size of these particles by simply changing the microbe we use. So what we see here, these are just different pictures of the different crystals you can get by simply changing the microbe. This is with direct air capture in minimal media, so it's a very inexpensive process. And with that, we can control the physical properties. And so along with that, what we've actually been able to do is to make building materials. So using 0.1 weight % of microbes to cement, we basically mix dried microbes in the cement mix and set it. And these pucks are 10 centimeters by 10 centimeters. So they're pretty large. And then we just destroy them with a hammer. And if we keep them in a humid environment for a little under a month, we get about 40% of that crack healed. And we can increase that percentage of microbes to speed the crack healing, but we don't lose at 0.1 weight % any of the compression strength of that cement. So what this means is we can use this for building materials and maintain the structural properties we need, while also decreasing the cost and time associated with repairs. So I went through those examples pretty quickly, just because I know this is not necessarily the area that most people here work in, but I hope what I've shown you not only is, as we think about sustainable technologies, how do we report on data in a consistent way, something to think about for tomorrow as well, but also how we can think about using what nature has already provided us to make technologies that are inclusive. What we mean by that is inexpensive, easy to access, and equitable. So I need to thank my team, and we have plenty of time for questions, but I didn't want to keep you from lunch. So yes, happy to take any questions.